Metal and metalloid toxicity. Elements like copper, iron, zinc, nickel, cobalt and aluminium, micronutrients, can be toxic to plants. Essential heavy metals are required in small quantities for vital functions. When present above threshold limits, these elements interfere with metabolic pathways, affecting pigments and enzymes. Uptake and accumulation occur through physiological and molecular mechanisms. Heavy metals and metalloids play roles in plant development as micronutrients. When concentrations exceed thresholds, they become toxic. Elements like arsenic, cadmium, lead, and chromium have detrimental effects. Aluminium toxicity is linked to acidic soils. Bioavailability and pH. Soil pH affects metal bioavailability. Some elements are more accessible at pH less than or equal to 7. Metallic ions compete with essential ions, disrupting enzyme functions and increasing oxidative stress. Aluminium, copper, lead and zinc readily bind to cell wall pectins. Plants are classified as hyperaccumulators, 1000 μgg, or non-hyperaccumulators, 500 μgg, based on metal concentrations. Strategies for damage control include sequestration, exclusion, chelation, and speciation. Constitutive transporters play a role in the active transport of divalent cations, and trivalent ions can also be taken up by specific transporters, NRAMP. Once ions cross the root barrier, they can be translocated via the phloem. Various transporters contribute to hyperaccumulation. Transporters like zinc-iron permease, ZIP, and heavy metal transport ATPase, NRAMP, CDF, and ABC are crucial. They introduce chelated metals metalloids into plant cells and translocate them to accumulating organelles, cell walls, vacuoles, in leaves. They mediate UDP glucose efflux into the cell wall, modifying its composition. This reduces the aluminium binding capacity to the cell wall. Studies in Andropogon virginicus reveal ABCG transporters' presence in roots and leaves. These proteins are found in cell membranes, root endo and exodermis, and vascular bundle sheath and epidermal cell layers. Their role may involve aluminum accumulation in plants. Phe-LS3 gene and abscic acid. Phe-LS3 gene expression increases with higher aluminum concentrations. Magnesium competes with aluminum for binding sites within root cells. Aluminium. Under acidic conditions, pH 5.0, it becomes solubilized as hexaqualuminium. Aluminium toxicity inhibits root elongation and interferes with nutrient uptake, limiting plant growth in acidic soils. Aluminium induces stress in roots, leading to anatomical changes. Modifications occur in xylem vessel length, diameter, and metaxylem vessels. Stomatal closure reduces photosynthetic activity. Accumulation in specific plants, example common tea, hydrangea, buckwheat, can accumulate high aluminium concentrations, 1000 to 3000 mg/kg or higher. Toxic effects of lead. It affects plant growth, development, and overall health. Symptoms include stunted root growth, necrotic lesions, leaf chlorosis, and reduced seed germination. Photosynthetic machinery is disrupted, leading to decreased energy transformation efficiency. High lead levels stimulate reactive oxygen species production, impacting plant health. Sensitive plants experience leaf modifications, thylakoidal membrane disorganization, and chloroplast morphology alterations. Resistant plants show changes in mitochondria structure and nuclear membrane disruption in root cells. Cadmium toxicity, it is highly soluble and accumulates in soil. It poses a risk to plants used for human or animal consumption. Higher cadmium concentrations are toxic, especially in roots. Arsenic toxicity and forms. Arsenic is a non-essential metal compound resulting from human activities. It exists in three main forms: arsenite, 3, arsenate, 5, and methylated forms. Uptake mechanisms differ for each form. Both forms cause oxidative stress. Root uptake and translocation. Arsenic is taken up through plant roots. Roots and tubers accumulate in high amounts. Reports suggest that not all arsenic is translocated to shoots, and edible plant parts may not accumulate it significantly.